Hi there, my name is Aaron Gupta, and today I will be presenting to you about a sender-receiver model to recapitulate multicellular patterns. Pattern formation has countless examples across a wide range of phyla. Many of these examples are in development, which I will go over later. They usually involve cell-cell communication via autocrine, paracrine, or endocrine signaling. And these patterns are often a result of chemical gradients, and oftentimes those chemical gradients are small molecules. One example I came across in the literature was the development of the neural tube in the mouse embryo. The partitioning of the neural tube is very much influenced by chemical gradients. As you can see here, three ligands, Sonic the Hedgehog, BMP, and Wnt, all influence the neural tube development. Now the goal with my project was to design a system that mimics this type of pattern formation using one-way communication between sender and receiver cells. Furthermore, I wanted to discover which kinetic parameters of my genetic device influence the timing, sensitivity, and stability of the system. To do this, I used a sender-receiver model. A single sender population will propagate a small molecule signal. Then, a heterogeneous receiver population will pick up that signal and respond to it based on the signal strength. Now, signal strength could be determined by a multitude of factors, but I'm most interested in the distance between the receiver and the sender. You can imagine on an agar plate that the signal would diffuse across the agar, and receiver cells that are farther away from a sender cell will pick up less signal than those that are closer to the sender population. And it is the kinetic parameters of each receiver subpopulation that determines the degree of their response. So the expected behavior I would have would be a single sender population, as you see on the left, and a multitude of receiver subpopulations, which fluoresce as a reporter based on their distance from the sender. You can imagine that if a receiver population that's heterogeneous surrounds a single sender population, that you could get some type of bullseye pattern if certain subpopulations are tuned to only pick up an intermediate range of signal. For example, the blue receiver population cannot fluoresce if it is too close to the sender population or if it is too far from the sender population. But if it is in an intermediate range, then it will glow blue. The same goes for the green, red, and yellow subpopulations. The genetic implementation of this system is really quite simple. A sender cell constitutively produces a small molecule, which I'll call M in this case. It exports that small molecule, which then diffuses towards receiver cells. The receiver cells constitutively produce a receptor for that small molecule ligand. In this case, I'll call that R. That small molecule and receptor form a dimer, which we'll call the MR complex. The MR complex then drives the production of two repressors, R1A and R2. Furthermore, R2 represses R1B. Finally, R1A and R1B repress GFP. This will be made much more clear on the next slide. So basically, we have a genetic circuit in which receivers act as bandpass filters, in that they only respond to signal if it is in an intermediate range. If the signal is too low, they will not fluoresce, and if the signal is too high, they will not fluoresce. Here, I have diagrammed the genetic circuit. MR drives the production of R1A and R2, R2 represses R1B, and both R1A and R1B repress GFP. The top part of this circuit is what we'll call the high-pass component of the bandpass filter, and the bottom part is the low-pass component. That's because if the signal is low, it will not drive the production of R1A or R2, and therefore R1B will repress GFP. Similarly, if the signal is too high, R1A will be in abundance and will repress GFP. However, if the signal is in an intermediate range, R2 will repress R1B, and R1A will not have the strength to repress GFP completely, and thus we will get fluorescence. Now it is very key that R2 is a much stronger repressor than R1A, since MR drives the production of R1A and R2 equally. In order to observe this system, I used a continuous differential model which considered 7 dynamic species in 14 biochemical reactions. Now, this system is generalizable to account for multiple small molecule signals. That's why I just use the signal M, and my genetic constructs are relatively abstract. In my model, I made a number of key assumptions. First, the diffusion of the small molecule M in and out of cells is instantaneous. Second, mRNA levels are at steady state and are not considered in the differential model. I am only looking at protein levels. Third, the MR complex degrades without dissociating. For more information about my model, its biochemical reactions, and differential equations, you can consult the PDF which I have attached with this submission. Many of the constants that I use are from a paper written by the Weiss lab in 2005. This graph shows the relative levels of all 7 dynamic species in the system. In this example, I pulse in a greater and greater amount of signal every 1,000 seconds, which you can see on the x-axis. When the signal is low, there is high R1b, and as the signal gets greater, we get a spike in GFP when R1b levels decline. Then, as the signal gets higher and higher, we see that R1a levels go up and repress GFP output. In this graph, I'm considering the steady state levels, when I pulse in a certain amount of signal and hold it there constantly. Here, we only get a GFP response when the signal is in intermediate range. Thus, it shows the bandpass nature of each receiver subpopulation. Here, we see that similar bandpass behavior, as concentration is on the y-axis and time is on the x-axis. The bandpass is optimal around 10 to the negative 5th to 10 to the negative 4th. However, on the ends of the bandpass range, we see significantly more time delay, which means the receivers are slower to respond to that level of signal. Now, this slide really shows the meat of the presentation. This has 2 receiver populations and thus is a heterogeneous receiver population. Now, the differences between these populations is that they have different bandpass ranges and accept different levels of intermediate signal in order to fluoresce. Here I've labeled them P1 and P2 for population 1 and 2. And the main differences between them are Kcat, which is the rate of MR dimerization, the alpha R1a, which is R1a protein synthesis, and alpha GFP, which is GFP protein synthesis. And when those are all smaller, you get a higher bandpass range, which is characteristic of P1. So if P1 and P2 had different fluorescent reporters, they would glow differently based on their distance from the sender population. Here I'm considering the transfer curve from off to on state. And when I was doing this analysis, there was only one parameter that really stood out to me, and that was the R1b degradation rate, or gamma R1b. Since R1b represses GFP when the signal is too low, as the signal gets higher, R1b will degrade and GFP expression will occur. However, if it's too slow, then it takes longer for that GFP expression to go, since R1b represses GFP for a longer amount of time. So from this model, I have a few conclusions I can come out with. Receivers with different bandpass ranges primarily different in rates of R1a and GFP synthesis. Fast R1a synthesis allows a quick switch to the off state as signal goes too high out of the range. Similarly, fast R1b degradation allows a quick switch to the on state as signal goes from low into the intermediate range. And when the signal reaches a steady state, the fluorescence does as well. This is true of both the on and the off state. Now, if I were to continue work with this project, I would definitely want to try to expand the receiver bandpass range. As you can see before, when I showed you the bandpass plot, it looked like a negative parabola. Now I'm interested in expanding that parabola so it's wider, so a receiver can accept a broader range of input signal levels. I would also want to turn this into a spatiotemporal model in which senders and receivers are various distances from each other and receivers respond to sender signal based on that distance. I would also want to include mRNA dynamics to see how they influence the total system behavior. Finally, it would be nice to create a surface plot that would model the NAND gate behavior of the R1a and R1b repressor. Overall, I think this type of modeling and analysis would prove to be very useful in analyzing natural processes such as embryonic development.